Welcome you all to the next lecture on inorganic chemistry of life, principles and perspectives. In the previous class, we have been looking at the oxygen transport system. Towards the end, we were trying to look at the oxygenation curves difference between the myoglobin case and hemoglobin case. Just let us recapitulate that one and look at this. The myoglobin case is completely hyperbolic, and if we were to have a protein which should have an optimal binding as well as the transport property of this, it should have a transition kind of a model, and that transition model will lead to this what you have here is a sigmoidal, and if you compare the hemoglobin curve is exactly like a sigmoidal kind of thing. So, you have a weak binding, you have a strong binding, reason there is a transition. So, therefore, the myoglobin is can only hold the oxygen, whereas the hemoglobin can pick up as well as the transport. So, now look at various events that we studied regarding this one. So, first thing is, let us say you bring the oxygen and bind it to the one of the subunit. That means, when O2 binds the one of the subunit, what will happen? We have seen already the iron 2 plus goes from high spin to the low spin, and then iron 2 plus size contracts, and then it goes more into the plane of the porphyrin ring that we have already seen. It goes more on that. But then what will happen? There is a histidine attached to it. So, obviously, as a movement from it, which was about 0.6 angstroms above the plane, now after the oxygen is bound, it comes to about 0.2 angstrom above the plane, but means more or less into the plane. That will disturb the connectivity that you have, or it will move the histidine bond that is attached to the iron. And that kind of a movement in that will trigger a conformational change. That conformational change go through that particular protein and to the neighbor, translated through the hydrogen bonding network, and then that will enhance the ability of the other units, neighbor units, either to bind to the oxygen. And this is the kind of phenomena. What we are saying is, when one oxygen binds at one unit, the binding of the oxygen, binding affinity of oxygen at the second, third and fourth becomes absolutely very high, and therefore, such a phenomena you can call it as a cooperativity effect. This effect is happening between the one heme of one of the subunit to the other heme of the other subunit to the other heme of the other subunit and the fourth. So, all this. So, and similarly, when it releases also, it goes exactly the reverse way. When one mole of one of the oxygen is released, then the rest of the oxygen from the rest of the heme centers will also get easily released too. So, the oxygen binding, then it contracts the size of the iron gets more into the porphyrin ring, that will give a kind of a movement on the iron histidine bond, and that will create a conformational change, and this conformational change is transferred from one unit to the other, and which results in the cooperative effect. I hope now you understand how the oxygen is cooperatively bound in case of hemoglobin, and this is what is the responsible. There are many other facets to this, how much of the conformational change, which region of the conformational change, these are all not part of this particular course. That will be something comes from the biophysical aspects, etc. But we just say that there is a conformational change of the protein moiety, because the iron is coming into the close to the plane, and then iron to the histidine bond is moving. So, we have looked at aspects corresponding to oxygen binding, what kind of a protein characteristics should it have. It should favor the oxygen complexation, it should not favor buoxodimer formation, it should not favor electrons transferred into the O2, and all these kinds of things. Then it should have both the binding in the weak to the strong by transition, having a sigmoidal kind of a oxygenation curve, but not the hyperbolic kind of a oxygenation curve. So, all of these are perfectly valid with this, and therefore, you have a cooperative effect of these the oxygen transport in this. Now, look at one another aspect. There is a lot of demand for synthetic blood. You know why? Because a lot of patients have to be injected, a lot of the leukemia patients have to be refilled, remove all the the old blood and refill with the good and fresh blood, etc. So, therefore, there is a huge demand for synthetic blood. So, there are some efforts in the literature. There are several efforts in the literature to go towards this. Just look at this particular thing. This entire thing is a protein, and you have a heme. This is the oxygen, you can take it as. So, it is basically a heme. The oxygen and the heme is buried inside the protein, and as a result of that, the mu oxodimer is not formed. As a result of that, it is able to perform many things. Similarly, people have looked at, can we make some kind of a mimic to this? So, what they done was, this is just the simple porphyrin is connected to the histidine, okay, and of course, there is a O2 here, and then on the periphery they have built certain units, so that it is a kind of a fence. So, this is a fencing around that, so around the O2, and in fact, even interaction is also being given over there. So, that means this FeO2 would not go to form FeO, Fe kind of a mu oxodimer kind of thing. So, people have, there is a huge number of different developments in the synthetic porphyrin, synthetic heme chemistry, but we cannot go into all of them. Just I brought to your notice that the synthetic chemists are smart, and they have tried to mimic this because it protects this one. They also try to protect by using this fence, and this is how the picket fence kind of a structures have come. Of course, still we have many other aspects are required to make this as a synthetic blood, a bit not so immediately close to that. So, let us not too much worry about, but efforts are always on towards the, making the synthetic blood. Okay, now we have looked at more or less exhaustively as a biological and organic chemist need to know the role of the iron, the role of the heme in a myoglobin and hemoglobin. Now, let us look at the other ones where you have a hemerythrin in invertebrates and hemocyanin in mollusks. Let us look at this and you see that this is the total protein, and protein is stretching 1, 2, then 3 and then 4. It is a kind of a tetramer you have. So, it is a tetrameric kind of a protein, and in each of the unit you have this di-iron center. Okay. So, in the hemerythrin you have a di-iron center. One of the iron is 6 coordinated, other iron is 5 coordinated, and this has 3 histidine, this has 2 histidines, and these are bridged by the carboxylates of one is aspartic, another is glutamic, and then there is a bridging. The bridging is not O, basically OH, and how this OH shuttles I will explain when we go from deoxy to oxy. So, this is a deoxy form as such, then how the oxy form, etc., then we will look at just in a while. Okay, so, in this particular case, the deoxy form, you have 2 iron centers, both the ions are 2 plus, and there is a bridging is OH. Of course, these bridgings are besides the carboxylate bridging, carboxylate bridging is there, but in addition. Now, in case of oxy hemerythrin, in the case of oxy hemerythrin, the irons are oxidized. As I told you in the beginning itself, you can see the 2 ions becoming 1 electron here, oxidized 1 electron, totally 2 electrons. Obviously, you should think the 2 electrons must have gone somewhere. Where they have gone? They have gone into the O2, and once they go into the O2, it becomes O2 to minus, and the OH transfers the proton. The OH the bridging OH transfers the proton, that will become OOH, peroxy kind of a thing. So, oxidize. Now, you can see there is a huge difference between the oxy form of hemerythrin and the oxy form of hemoglobin. In the oxy form of hemoglobin, nothing happens to the oxygen bond order to not much extent at all, whereas in the hemerythrin the oxygen OO bond order is reduced. This is all I explained to you. Okay. So, this is besides the carboxylic bridge, etc., etc. So, therefore, those are something important. Now, so is a di-iron center. So, one can make some kind of a guess between the two iron centers, what different ways a O2 can bind. So, one other thing it can be binding like this, or one other thing it can be binding like this, one other thing it can be binding like this, and one other thing it can be binding like this, or it can binding to only one iron, but like this. They are possible. How will we know these things? How will we differentiate? Can anybody think of? What are we looking at? We are looking at the way that the OO is bonded to either one iron or both the irons. That means we need to get a click in our mind that we can look at the OO stretching vibrations. That means vibrational spectroscopy or infrared spectroscopy or FTIR will be able to tell us whether the binding mode is like this, whether the binding mode is like this, whether the binding mode is like this, etc., etc. These have been done with a small molecule and they have been compared with the protein systems. So, we are not going into all those details and ratify. All that I want to tell you is the OO stretching frequencies differ if you go from type 1 to type 2 to type 3 to type 4 to type 5, and that itself is a good message for you to say that yes, this is the FTIR spectroscopy can be used. Okay. This is not so much of importance here because I will be explaining this later, but since that binding modes are there, I have shown it over here. Okay. So, what is happening here? The ion which has a 5 coordinated will bind to the OO and both the ions will give 1 electron each, then OO minus, and the OH hydroxyl proton is transferred to this. In fact, it is stabilized by some kind of a hydrogen bonding interaction as well between that oxo bridge and this OOH part of it, oxo bridge and OOH part of it. So, this is stabilized. And now, so as I said, since this is possible to be differentiated from that, so we have now here vibrational spectrum in the new O2. But this vibrational spectrum is not used just to meant for differentiating all the 5 forms, and this is used to meant to identify whether the kind of a binding is in this particular mode or not. So, therefore, just to identify that it is in the form of the species 4, type 4 species, that is what we have. And if you look at the OO stretching region, you will find the OO stretching vibration. Now, you can have a natural abundance of OO, then 16 O2, or you can partially change into 18 or completely change 18. So, as you go from 16 to 16 and 18, there will be a frequency shift, and frequency shift will be in the lowering side. And you go to the 18, the frequency shift will be more than lowered. Why? Why the frequency will lower? You know, in the infrared spectral stretching vibrational frequency, on the numerator you have a bond order or bond strength or force constant, and the denominator you have a mass unit. Here the mass is OO mass. So, in one case 16 mass, reduced mass you have to find, that is 1 by 16 plus 1 by 16. Or 1618 or 1818. So, obviously their reduced mass are different. So, therefore, you find peaks accordingly. We do not need to analyze much more than this. Okay. So, therefore, we can try to understand that very nicely, and let us look at the process in a simplistic manner. because this protein is a very tough protein, it is a huge protein, and studying all the phenomena is not that easy. So, we just take only comparative kind of a approach here. Now, you see that the ion A is having 3 histidines, ion B has got 2 histidines, and in the deoxy form both are 2 plus, and the bridging is OH. And one of the carboxyl bridging from aspartic acid, other carboxyl bridging is from the glutamic acid. Perfect. Now, if the O2 comes in proximity, where will the O2 bind? Because there is a vacant site here, the O2 will have an interaction over here. So, and when the O2 binds, there is an activation, and both the ions will give 1 electron each, and then this is what is formed, and this one will have, this is the OH plus, and that will form a kind of a hydrogen bond. You see that bridging oxo, the hydrogen which is present here has transferred to this, and therefore, you form a kind of a metalla cycle, ion OH O cycle. So, it is a 5-membered metalla cycle that you have formed here, and this will have obviously a different kind of a OO stretching vibrations, OO vibrational spectrum as compared to the other modes that I have shown on the earlier one. That is how we are able to show all these things. So, here we have not gone into so many details because these proteins are very huge. So, therefore, we have restricted ourselves strictly to the metal center, and then we try to understand that phenomena. Now, for a while let us move to the third case, which is the hemocyanin. So, the hemocyanin from mollusks and arthropod, it is blue blood. Okay. So, let us look at the protein is also huge here, not that simple, okay, and this is a quaternary kind of a structure. Now, inside each of the subunit you have a 2 copper centers. I think these 2 copper centers are farther apart by about 3.5 angstroms or so, 3.5 to 4 angstroms or so, and each one of it is 3 histidines, here also 3 histidines. You see that 3 histidines here, 1 histidine, 2 histidine, 3 histidine. Similarly, 1, 2 and 3 here, and the copper is in copper 1 form. This is called the deoxy. Now, you add the O2 to this, and very instantaneously it will form a kind of species like this. How do we know? From the, again from vibrational spectra you can make out the way that it is being binding. So, vibrational spectra is really an instrumental and key type of a method to identify the way that the O2 is bound, in what manner that it is bound. So, this is the important aspect. Now, here what happens, copper 1 will go to copper 2, there are 2 electrons in peroxo. So, here the peroxo is bridged very symmetrically here, and in the previous case the hydroperoxo is not bound symmetrically. This is an unsymmetric peroxo, and this is a very symmetric kind of a peroxo. Therefore, your vibrational spectrum will differ, and that is how things are formed. Now, let us look at all the cases. If you look at the hemoglobin, then you find stretching vibration around 1200 or so centimeter inverse, and hemerythrin around 850, and in the hemocyanin 750. This is the O2. O2 has a bond order 2, gives around 1500 range the vibration. Remove 1 electron from this, becomes O2 plus, bond order should increase, because where are you, why the bond order should increase? Because you are taking 1 electron out of the antibonding orbital. So, what is bond order? Bond order is number of electrons in the bonding orbital minus the number of electrons in the antibonding orbital by 2. So, since you have taken out 1 electron from the right side subtraction, which is the antibonding orbital, obviously you are increasing the bond order, and therefore your frequency will go up. Okay. On the other hand, you put 1 electron. When you put 1 electron, what will happen? This electron will go into the antibonding orbital, and the antibonding orbital has more electrons, then the difference will be less, by 2 it will be 1.5. So, therefore, the partially reduced, and then if you put 2 electrons, it is further reduced. So, 1.5 electrons around 1100 kind of thing, or a bit lower, and this is around 850. Now, if you compare with this There is the least perturbance in case of oxygen bound to the hemoglobin iron, and there is a intermediate perturbance when it bound to the hemerythrin. As you can see from the structure, it is not centered between the two irons. And when you go to the third case, hemocyanin, you can see the OO is exactly centered, is exactly binding to both the coppers, and that is what is happening in these cases. So therefore, that will be making much more reduced form, and that gives the 750. So you can see that the O2 in the hemoglobin is not that much perturbed. O2 in case of hemerythrin is quite much perturbed, almost about 1.5 kind of thing. And then when it comes to the O2 2 minus in the hemocyanin, it is very much like the O2 2 minus, perfect O2 2 minus, and that is what we find. Now, whatever I said till now, let me summarize putting into these one. This is the hemoglobin part of it, porphyrin and center metal, and this is the guy which is the proximal directly bonded, and this is the distal which will recognize the substrate, and it binds FeOO. Now, you come to the hemerythrin, there are two O ions, there one iron here, one iron here, and in absence of O2, these are all in iron 2, but once the O2 is approached, both the ions will turn to iron 3, because this iron 2, iron 3 can be differentiated by using EPR, Moss-Boyer spectroscopy. There are a lot of studies that are done using EPR and Moss-Boyer spectroscopy, wherein you can identify not only the ion, whether it is a 2 plus or 3 plus, and you can also identify whether the ion is in the low spin or high spin. So therefore, using the combination of the Moss-Boyer and magnetic aspects, EPR, one could fully establish, and these have been done in this. I am not taking the details of this into this particular course, only I am taking the results out of that and using it. And here it is bonded to one of the irons, and therefore it is not symmetric, and therefore the entry of the 2 electrons is not as good as in the case of hemocyanin. See that hemocyanin, and there are 2 coppers, and then you have the O2 here. It is exactly symmetric, and it looks of that kind. These are this kind. Of course, this is not linear, this is bent, and this is like this. So all of these can be very nicely studied by the vibrational spectroscopy, and the oxidation states and the spin states of the ion can be studied by EPR, can be studied by Moss-Boyer and magnetic measurements. So one should be able to answer these things when we have some questions coming in this particular direction. So that takes to the end of the oxygen binding, oxygen transport in higher organisms like human, and the lower organisms and much lower kind of invertebrates and the mollusks kind of a thing. So one iron in the human system, 2 ions in these ones, and in the mollusks you have the 2 copper centers. Okay. So therefore you can see all of them. Now let us move on to the transport, but not of the oxygen, but transport of some ion, let us say. So transferrin is an enzyme which transports the iron ions, because iron is required in the entire body for a variety of cellular functions, and this is being transported by a protein called transferrin. And this transferrin is a blood protein, and it will take to the different organs of the body and delivers there. So therefore, the iron based enzymes in turn are enriched in the cells of each of the organ of this body, and that is what is going to be the important for the functioning of each of the organ. So let us look at the ion transport, general aspects of it. General aspects of the ion transport, there are different common proteins like transferrin, ceruloplasmin, albumin, phosphoprotein, and there are some directly it can go without even ions like iron, ions like copper, zinc, calcium, variety of these ions. In fact, many more are there. This is just a few. and their cellular or extracellular concentrations are, some of them are extremely low, but they are very important. 10 power minus 16 molar, 10 power minus 14 molar, albumin 10 power minus 14, okay, and let's say, and phosphoproteins, and so all these kinds. So these are proteins are present in our body, in the blood, and in the blood these ions are picked up and then transported to various organs in the body. Particularly we will look at only one example, that is the transferrin example in this. Okay. So, just before going into the transferrin story, let us look at some more general features of ion transport, particularly in a small organism like E. coli. So, look at this, you have an outer membrane and you have an inner membrane. Now, iron comes as an iron chelator, iron complex, etc. Okay? And this is transferred at the surface to these iron proteins, which are called iron uptake protein, ferric hydroxamate uptake, transport of iron, TON. So, all of these. So, it is transferred to this, and these in turn are carried and transferred to this inner, and this further goes and releases in the blood. Okay. So, you have a one level of capture of the iron, transferring to another level of things, and then transferring into the blood. Okay. The outer membrane and the cytophoreal complexes facilitate the movement into the cell. Thereafter, a variety of transport proteins. So, these kind of proteins are all there, and they take it into the cytoplasm. Okay. Or in this here they will take into the cytoplasm, and that is an important aspect to be learned. Now, let us look at the transferrin now. Transferrin, as I said, a protein looks like this, a huge part here, a huge part here. So, one of them is an N terminal, other is a C terminal, and there is one iron center here, there is another iron center here. And if you look at the iron center, in both the cases they are very much similar, whether it is a N terminal or C terminal. The only difference is that their binding strengths. C terminal is bound a bit more stronger than that of the N terminal iron ion. This is one thing we need to make. Okay. So, this is a crystal structure, and see very close to the iron, you can see that what is bound over there, and this is nothing but these ions, groups, tyrosine, okay, and these are the two tyrosines and the histidine. And in addition, you see something like, this is not a part of the protein, but these carbonate ions are very essential, found to be very essential. If the carbonate is not there, the enzyme is not complete. So, I will explain just in a while that one, and so therefore, for an apoprotein, iron and the carbonate are required to form the whole enzyme, but without the carbonate it will not. So, therefore, the carbonate is a kind of a allosteric factor in this, but essential factor in this. So, you can see crystal structure from this, and if you look at an apo form, you see that the tyrosine, histidine, imidazole, etc. Then you have. So, this is a kind of a core where the iron comes into picture. Okay. So, this is an interesting phenomena that one can look at. Now, you take, as I mentioned, the transferrin apo form. You take the apo form of the transferrin, start adding the iron to that. So, if you start adding iron, you can do iron as an iron 2, you can do as in presence of the air, or you can do the iron 3 also. So, let us take this one of the apoprotein of the transferrin and add iron to that. So, if you start adding iron, different mole equivalents of the iron, you see the absorbance at around 275 nanometer, slowly increasing and then saturates, and it intersects at this particular region. So, this particular region is basically responsible and explains that there is a complex formed between the iron and protein is what? Here you can see that intersection is 2. And why 2? Because there is one iron center at the N terminal, other iron center at the C terminal, therefore 2. So, you are getting 2, absolutely right. You can also get this spectra at different pH for the transferrin and presence of the apotransferrin as well as in presence of iron, and these are what is shown over there. You can see a very nice complex is formed. This is transferrin at pH 6, iron transferrin as at pH 6. Similarly, transferrin, free transferrin at 11 and iron with the transferrin at 11. So, you can see very nice bond, and in fact, you can do a very nice titration and try to find out pH variation also. You can find out at what pH the complex is stable, etc. But this all becomes only the additional things, but no new information. So, the information that we get in this is that it binds in the 2. So, when it binds the iron 2. If suppose you add transferrin, carbonate is essential, as I told you. It will form initially iron 2 and then get oxidized to iron 3. Or you can take the apoprotein and add iron 3 as well, and then this iron 3 will be straight away, no need of oxidation. So, the apoprotein if you take and you can use the transferrin, the apoprotein transferrin and add the iron. So, therefore, the iron is basically added. If it is iron 2, initially the iron 2 complex forms and then followed by in the presence of oxygen, oxidation takes place, or if it is iron 3, directly you have a iron 3 bound thing. So, as you can see that we have a system of the iron transport system, which is transferrin having 2 centers of the iron binding with the iron at the C terminal and the N terminal, so we could see very nicely how the iron binds, how we will find out iron binds, how many ions are bound. We could see all of them. Now, in the next class we will look at how this particular iron is being transported and what are the other characteristics of the iron transport protein that we will see in the next class. Thank you very much.